So can you see my slides?
Yes.
Yes. Okay. So good afternoon, everyone. So welcome to the seminar. First of all, thanks again for the introduction by Professor Choi. So I am Joachim. So you may call me simply Along or in 普通话 Alang. So I would like to give a brief introduction of our newly formed single-cell omics core, in short SCOC. So the talk contains several contents, namely, there's a general overview of that, how exactly we going to work on that, the workflow. So the ways how to prepare the single cell suspension for the experiment, the single cell preparation part. And I will give also a little bit introduction on the SCOC equipment that we have got. And lastly, we will, if we have enough time, then we will have a brief introduction on the sequencing recommendations, as well as some data analysis part. So lastly, let's begin. So the idea of the single cell RNA sequencing came out from almost more than 10 years ago. So in 2009, a paper generated from Deng Fuzhou et al. So lately, he became the full professor and expert of this field, and proposed a paper of a cherry picking single cell partitioning of and to profile this genomics and also transcriptomic profile of the single cell. But at that area, during that time, sequencing is a very expensive task, and not everyone or lab can afford to make an test sequencing of single cells. So that time, that was not a major way of looking at the transcriptome. So if you remember that correctly, during that era, most of the people will address the transcriptome in a box sequencing way. So in terms of to tackle the expression profiling of the box sequencing. So in term meaning, so the expression profile of the samples in as a whole. So you may not know the expression profile from the individual cells. So as time goes on, so more and more groups and people and targeting on the technical hurdles on how exactly they can tackle single cell resolution. So in 2001, so they start to cherry picking single cells into the 96 well plates, and then to multiplexing them by putting a small oligo, which is an individual in cell by cell manner. So, and then after that, they mix them together to identify cell by cell by in terms of transcriptomic profiles by the multiplexing oligos. So, but still, the throughput was still very, very low. So until the year 2013 and further on, then the development of these microfluidic devices greatly advanced the development of a single cell RNA sequencing. So, so as you may know from the figure B at the bottom, so in the middle, in between 2013 and 14, so this Fluidigm C1 machine emerged. So by using this Fluidigm, this microfluidic devices, then the PIs can greatly induce and single cells into sorting into the single partitions. So at that time, so a single experiment can be easily be done with 96 individual cells. But at the same time, so a more conventionally, commercially available techniques or chemistry, such as the SMART-seq and also the cell-seq developed. So by this technology, then it is available for the PIs to generate full-length transcripts from the single cells. So this is not still at the era of a highly throughput RNA sequencing until the emergence of the drop-seq or the in-drop technologies after in 2015. During that time, then people came up from an idea that what about using oil droplets as a media to set up a physical partition for a single cell, meaning so in a single well, then you can simply put a numerous oil droplets, each having 0 or 1 single cells. So you can treat that as a single droplet or oil droplets equals to a single partition of a traditional 96 well plate well. So by doing so, then people largely increase the throughput from just 96 cells at a single experiment to thousands or even more cells at a time. So in 2016, a major step through that 10x Genomics, the company established, and they propose a chip-based, highly throughput single cell platform. So which this SCSC is adopting this the chemistry from this company with a droplet-based encapsulation of single cells as a partition for the single cell RNA sequencing. So in 2006, they developed a droplet base. They make it as a gem. So in short form for gel emulsion, gel bits in emulsion. So you can, from the name, so you can figure out that basically it uses the in-drop or the drop-seq chemistry. So but they are making some further modifications on that. So you can see from this diagram that from a single tube with a different oil droplets, and then you simply load the cells and together with the oligos, which I will explain in a minute, that together, and then so they mix it into a droplets, each containing a cell together with the RT, enzyme, and reagents, and also with a gel bit with the oligos, which I'm going to explain a little bit. And this is just a introduction of the general methods or chemistry 10x Genomics is using. In talking about that in further, so basically this 10x Genomics uses a major component of this kit. It's called the gel beads. So if you magnify a single gel bead into, so magnify it up, and then you will see there are numerous oligos being conjugated onto the surface of the gel bead. So if you focus on a single oligo, then you can simply see a simple structure containing a poly T tail, which can conjugate with the poly A tail of a mature mRNA molecule, together with a UMI, which stands for a unique molecular identifier, which I will explain it further. Also with a fragment, which is called the 10x barcode. And another important component of that is a TrueSeq read 1, so which stands for a Illumina format read 1 sequencing adapter sequence. So which means this kit or the library finally adopted will be only available for the traditional or conventional Illumina sequencing platforms. So basically, this is an exact photo taken by 10x Genomics that how exactly one single cell is caught on inside the single droplet. So you can see the big circle is a oil droplet where a cell is captured inside together with a gel bead. So this is a gel that I was talking about with multiple oligos attaching on the surface. Coming to the workflow. So the workflow contains 2 full days of work, so which I will explain little by little bit. So in general, so the PIs or the users has to optimize their samples of input really well, and which I will explain again in further way. So the day 1 contains as I said, the first step is the encapsulation of single cells into the oil droplets. And after successful encapsulation, single cells will be lysed and RT will be conducted inside the single droplets. And after cleanup, then there will be a cDNA pre-amplification step, PCR step. So this is to, first of all, you generate the cDNA, and then the cDNA is multiplied or amplified a little bit to have a enough or sufficient amount for further DNA sequencing library preparation. So the two contains a shearing and also traditional DNA sequencing library preparation step, and also including a little bit of a quality control steps, and then basically then the sequencing libraries are ready for sequencing. So the first step, as I said, is the single cell preparation. So I would say this step is super crucial in the entire protocol. So meaning that if you have there are several questions to ask before considering a single cell experiment. So what I'm talking about is the single cell RNA-seq. Then for sure, then the stability of the RNA is the key. So how to approach that, the purity and the integrity of the RNA molecules, then of course, if you have the cells or the animals or the cell line fresh, then that will be the excellent example that or scenario that you firstly you consider the when or what you want to do. So are you going to work on the cell lines, or you want to work on the tissues from humans or from mouse? So what is the status of the tissue? Is it a fresh tissue or the frozen tissue? So is the cells or the tissues having biohazard problems, or simply if you have playing around with the virus transduction or so, then is the level 2 biohazardous safety issues and important issues to carry on. And further, there's your own experimental design, whether you have to, if you have to work on an experiment which is comparing a control versus a treatment group, or you just want to simply look on a single cell heterogeneity or the gene expression profiling of your target samples, then you would have to plan it in a different way. Again, so the sample amount of your target cells. So how many cells exactly you want to load into a chip or experiment at one time? So this is another good way to look on that. So I will explain the proper or optimal range of cell inputs in a minute. And also the tissue dissociation method, there are so many different tissues from human or from mouse. If you are playing around your cell lines, then you typically do a trypsinization, then usually most of the cell lines are good for processing. If you are playing around the tissues or so, then you have to figure out what is your best way of dissociate your target tissues into a single cell suspension. And if so, then what is your target viability or your numbers of your target cells? So if you have playing around a human tissues or the mouse tissue samples, then you better look on the viability. So if your enzyme is too strong so that you have a lot of dead cells, then whether you have to include a cell sorting process to purify your target cell tissues, or you just have to, there are some conventional available kits, such as the dead cell removal kits by Mutant Biotech or other companies, to simply remove the dead cells and purifying your target cell input. And then, so the next one is how exactly you are going to count your cells, whether you are going to count the cell by traditional hemocytometer, or you have other ways of counting. If it's your counting accurate enough, comparing your cell sorting with your hemocytometer human bare eyes counting method. So, which is also important issues to mention. And finally, so the Transcriptomics has an optimal range of cell concentration, which is a 400 to 1,000 cells per microliter. So if you have a cell line to load into the system, then of course you can have a lot of cells to start with. And also the cell purity is also good. But if you have to play with your tissues, which is having very, very trace amount, such as a rare tumor tissues from humans, or just a small tiny organ from mouse, from a knockout mouse string, then you need to think about your cell input. So it is not always that you can achieve that many cells at a time. So you need really to have to think about that. So so all these points from this list can significantly affect your efficiencies of your whole protocol. So it may have substantial effect on your data analysis and also your quality. So this is a simple way how to adjust or you assess your cell quality. So first of all, you have your cell state. So there are two ways to go through. So first of all, you need to ask whether your cells is a cell culture or the adherent cells, simply suspension cells. So if yes, then if your cells are having the concentration of, for example, 6 times 10 to the power 5, so 0.6 million cells or higher. So if yes, then you can go into this way, or no, then you have simply less cells, so you have to be suspended in less volume. And also and then you carry on to the cell washing protocol. So in general, the washing buffer for this step, then we always usually recommend a normal sorting buffer, or just a PBS supporting with 0.05% BSA. Sorry. So by just remind that each washing step can cause several extent loss of extra cells. So please be careful when you wash the cells. Do not touch the pellet, or you just have to spin down for a longer time for a significant obtain of your pellet in a good way. So after that, then you have to always resuspend your target cells into a proper sorting buffer or washing buffer, as I said. And so another important point is to place your cells always on ice, so but not in the ice. So I have previous experience of a case that you have a very little cells, so maybe just half a million or even less cells, and then they resuspend the cells into a very small volume, so that, and then they just put it into the ice, and then after half an hour, the washing buffer or the suspension just frozen. So then you lost all the cells. Then, so bear in mind that, so you have a small volume of a buffer with the cells inside, then just always put the cells on the ice. Yeah, this is just another reminder. So all in all, the cell preparation has a well-known or the well-established steps to get your cells prepared. And I'm not saying this is just a sole protocol for that, but there's so many different available protocols for your specific type of cells or tissues. Each user has to optimize your own, basically, the cell preparation protocols. So the general idea is to keep your cell as much as possible, and also your viability as high as possible. So if you have found out just a resuspect of cells from your frozen tissues and also putting on ice, and after several washes, and you have a perfect cell viability and also numbers, then congratulations, you have a perfect cells or material to load into the system. But usually it is not the case. What I mean is you have to optimize your protocol little by little bit. So yeah, for details, I'm welcome to talk about that. So here are some examples for the optimal and suboptimal single cell suspension. So from the left, then it is a photo taken from this cell counter from Thermo company, which is called a Countess cell counter. So you can see the dots are single cells. So pre-lysis, post-lysis, and also after washing and filtration. Then obviously you can see if you have putting an enzyme to segregate the cells into single cell suspension, you will see most of these cell clumps are gone, and some of these debris will produce. So after these washing steps, then usually we add up a filtration or straining by cell strainer. Usually we put up starting from 100 microns of strainers. Then after straining, then you can see really pretty clean background of a single cell dots. So at the middle column is the fluorescent signals of that. So you can still see the same pattern of single cells. So we welcome the optimal cell status at the upper panels. So as you can see, these are the single cell, suspension taken in a hemocytometer. So this, if you have clumps, then they will largely affect or even causing failure of the entire experiment. So as you may imagine that actually the whole, as I showed you before, that it uses the microfluidic way to pass through the cells or the nuclei into the microfluidics circulations. So if you have too many cell clumps, then usually they clot in the middle of the circulation, then they will cause a insufficient production of the oil droplets. So here is the actual, how the actual flow chip looks like. So there are 8 channels. So each channel composed of a different component, the number 1, number 2, and a collection well, number 3. And basically, you just have to load your sample, the cells, together with the master mix containing the RT enzyme and the reagents. And then you load the gel beads, which I explained before. So these are the synthetic gel beads with the conjugated oligos on the surface. And after that, then you simply load your oil droplets, your oil, partitioning oil into the well number 3. And by putting into the machine, then you can simply press, mix all these 3 together. And then so you will have the oil droplets stopping at the number 3 well. So as I said, there are 8 channels, which means up to 8 samples can be hosted per chip processed in parallel. So each well is suitable to put from 500 to up to 10,000 cells per channel. So which means if you have 8 samples in total, loading into the single chip, so but which means in a single day, you can just finish up to 80,000 cells per experiment, which is a super high throughput up to now. Of course, there are new other methods or companies working on a super high resolution or the super high throughput on that. But until now, Transcriptomics only can host up to 10,000 cells per well and 80,000 cells per chip at the same time. And then the machine can mix up all this together to generate oil droplets. And this process takes about 18 minutes. And as I said, so cell clumps are detrimental and lethal to the generation of oil droplets. That's why we prefer the PIs have to declare on and also to state that your optimal size of your cells. So we prefer to have to have a single cell suspension with the diameter less than 30 microns. So anything bigger than 30 microns, for example, if you have a large cell, such as a human oocyte or a hepatocyte or a neuron, then anything bigger than that, then you really have to treat that really carefully. So they may have a chance that to clot or to block the channels of the chip and cause a failure. Then if not, then usually there is a 60 to 65% of the cell recovery only from the total number of cells loaded into the system. Meaning, so if you want to aim a experiment with the 5,000 cells, then for sure you have to load much more cells than 5,000. So since there is some loss of cells within the microcirculation and also during the multiple steps of your sequencing library preparation steps. Here is a more detailed chemistry of the kit. What exactly it is doing. First of all, within each gem, so each gem means each droplet. So the single cell is sliced and the mature mRNA is released. So remember that this kit is only capable to catch mature mRNAs, meaning with the poly-A tail. So any other RNA forms which is not having poly-A tail will not be captured. For example, the ribosomal RNA, the tRNA, and also any other RNAs from prokaryotes. So, and also the microbes are not captured by this system. So all these mRNA molecules are released and then being captured by this poly-T tail of these oligos. As I explained before, so on each oligo, there is a UMI and also the 10x barcode. Then I explain it now how exactly it is working. So you can imagine that 10x barcode is the barcode of the cell itself. So all of these oligos having this from the same cell are supposed to have a unique 10x barcode. So meaning, any grabbed mRNA with the same 10x barcode will be assigned back to the same cell identity. So by this way, one can assign the unique mRNA molecule back to your original position, meaning to the original cell of contribution. The UMI itself is a marker for the mRNA. So meaning, as I showed you before, one gel bit contains numerous different oligos. So each oligos contains a unique UMI. So any mRNA being captured by this one will be labeled by a unique UMI. So imagine you have a 100, say for example, 100 different mRNA being captured in that cell, from that cell in that gel bit. And supposedly each mRNA will have a very different UMI. So namely 100 different mRNAs, 100 different UMIs. So this way to differentiate whether a molecule is a molecular duplicate or not. So if you can imagine that if you have a high expressing genes, such as for example, beta-actin, highly expressed cells of a normal cell, that usually you will have multiple transcripts available in the cytoplasm. Then if you grab all of the transcript together, then you have all of these transcripts will have different UMIs. Whereas if you have a rare expressed genes, then which may release only really a few different transcripts, then usually you will have all these transcripts will have a shared UMIs, meaning all these products being produced are molecular duplicates. So getting back to this protocol, so firstly, this mRNA is being captured by the poly T-tail, and the reverse transcription will be carried on as the reverse transcriptase has a relative template switching activity. So the original template will be swapped from this new oligo. So at the same time, this reverse transcriptase will add a extra Cs at the 5' end of the original template. So with the TSO, which means the template switching oligo, the template switches from this side to the real mRNA molecule. So after this one, then basically after this, when the RT is finished inside individual oil droplets, then we're supposed to have a double-stranded cDNA being generated. After that, then most of these molecules from mRNA molecules from the single cells are being labeled by the 10x barcode, the cell barcode, as I said. Also the UMI, the molecular barcode, as I said, for the unique mRNA molecules. Then you simply have to produce a second strand synthesis to have a double-strand cDNA molecule. And for this molecule, so at this time, it is no longer necessary to separate the cells into single cell partitions. So by this time, you can simply break the oil droplets into together and merge all these molecules together, as you have already your 10x barcode, the cell barcode, and also your molecular barcode being tagged. And after that, then we always have to do a simple QC on the cDNA being produced. Then we have a machine called a tape station bioanalyzer from the Agilent company that can quickly and efficiently tackles the proper sizing of your cDNA product. So this is a simple bioanalyzer electropherograph from Tennis Genomics that the peak generally rise in between 500 to maybe 200 and 2,500 base pairs. So the height of the peak depends on your initial amount of cells being loaded into the system. For sure, the more cells you put, then the higher the peak you have. And also the nature of your samples. So if you have loading a cell type, which is a very transcriptionally active, then usually you have a higher peak. If you're loading a relatively lowly expressing material, such as a white blood cells or PBMCs, then usually you will have a lower peak. And thanks to Professor Alfred Chen, which they have conducted the first ever trial on the PBMCs last week, then this is the real data, how exactly it looks on the cDNA profiles. So basically, as I said, you load in the PBMC into the system, and PBMC are the cell types which are having relatively lower transcription expression. So that's why the peak are lower. And also from the two visualizations of the bands, you can see actually the bands covers around 1,000 base pair. And the shape of the peak is relatively spiky. So which is a hallmark or a sign of a successfully produced cDNA. So the lower and the higher upper marker are the default marker from the system to marking the correct size of your band. And this 76 base pair band is actually the primer dimer. So the primer itself contains a maybe 35 or 36 base pair. And then so the dimer is actually a double size of that. So which is I would say a contaminant of that. So, but as the amount of that is relatively low, then I would consider this cDNA is having a high quality. Then after achieving this evaluation of the cDNA from the first day, then actually we can carry on the protocol for the second day, which is a sequencing library preparation. If you are familiar with the sequencing library preparation, actually this is just a replica of that. So from the cDNA, so as you can see, the medium size of that is around 1,000 base pair, which is far beyond the detection range of the Illumina sequencer. So the typical read length of the Illumina sequencer is around 200 to 300 base pairs. So depending on the running mode and also the whether it is a single end or the pair end mode. So as recommended by Tennis Genomics, then usually this long cDNA fragments are subject to enzymatic fragmentation. So as you can see here, the long cDNA is being fragmented into shorter fragments. That's why this particular step is unique. As you may know that it once being fragmented, then it is no longer a full-length cDNA molecule. That's why this surface or this kit from the 10x genomics is in general the 3' and RNA sequencing. So only the the reads is actually starting to be read from the 3' end of the cDNA. So depending on the length of the cycles that you are going to run. And in general, it is not a full-length RNA sequencing, but depending on your needs. So there are different, many other different protocols available. So, but in general, the 3' end RNA sequencing protocol has been shown or proved to be very efficient to map the transcriptomic profile of the single cells successfully, and in maybe the many other literatures and papers from the recent 5 or maybe 7 years. So I'm not saying that the 3' end sequencing is the perfect one, but in general, it can already tackle or address most of your questions. So if you need a full length of, such as if you have to profile or to address or to segregate your different splice variants from a rare transcripts, then the 3' end RNA sequencing may not fully fulfill your requirements. Okay, so after this fermentation, then most of these fragments are being fermented into a optimal size, maybe around 250 base pairs. And then after a conventional end repair A-tailing and adapter ligation steps, then you will end up with a molecule look like this one. So you have the real RNA sequence right here at the gray bars, and being surrounded by the read 1 and also the read 2 sequencing primers from the Illumina format. And after several cleanup steps and priming, there is a last step which is called the sample index PCR being performed. So since at that time, the total amount of the cDNA is still really low. So if you have to amplify your cDNA into a sufficient amount for enough sequencing, then usually you will have to sequence it maybe around 10 to 11 cycles. So at the end, you will have end up with a library look like this one. So first of all, you have the same cell barcode, the 10x barcode, the UMI, and also a sample index here. Okay. So again, so after successful sample library preparation, then again, you have to do a QC. Then again, so the SCOC here has the service to QC your final product. So again, thanks to Professor Alfred Chen, then they have already done their QC of their final library product. Then you can see from a spiky peak from around 1,000 base pairs, then so the final product looks like around a single peak, very smooth one, with a peak size around 410 base pairs, which is a very, I would say, very nice quality. So maybe we stop here first. That I would like to deliver some take-home messages at this point. So there are some other contents from this PowerPoint, but due to the time limitations, so I will maybe conclude a little bit here first, and then there are some other portions that I would like to share maybe in a later time point, or maybe during the Q&A section. So first of all is to plan an experiment well. So whether it is a necessary, do you have to run a single cell RNA sequencing experiment? It is not a cheap experiment. It is a sophisticated experiment. You have to optimize your cell really, really well. And please talk to me if you have any questions for a requirements of the cell input. And we have drafted the sample submission form for declaration of this sample nature. So to state that your cell is free of a biohazard matters such as a viral infection and so on, and that you have a high cell viability, that your cell is free of clumps, and thirdly, to submit your single cell suspension with optical quality and then concentration, of course. Then we will simply move on your experiment, and then we'll generate your sequencing library for you. And lastly, as I said, the surfaces is not including, however, not including the sequencing surface. So the surface contains from cell to the sequencing library, and the price is around, I would say it's lower than 20K Hong Kong D. So there is a discount for the SBS PIs. So for the detailed price, please come to me or come to Professor Choi or the director personally. So we will offer you discounts for SBS if you are a SBS user. So I would say we are doing a promotional price period at the launch period. And as I said, the package includes from cell to sequencing library preparation, and also for QC and quantifications. And if you need a extra help for advisors for sequencing platforms, such as what kind of a sequencing depth or platform on the Illumina sequencer I would like to go on, then I can simply give you some advices. So, but each sample or each cases will subject to individual discussions. So I would like to provide troubleshooting and also intellectual advices. So here I would like to welcome all discussions about your own projects. Okay. So last two slides. So I would like to show you the equipment that we have right now in the SCOC. So firstly, it's located at the room 525 of the LIBSB. And there are, as you can see, the cell counter, microscope, and centrifuge. These are for cell preparation. So I would suggest you to get your cells ready and contact me by, of course, you need to submit the online submission form, and then to talk to me which day exactly you are going to submit the cells. And then I will process the cells. If the cells are not having sufficient concentration, then I can simply prepare it nicely in the 525. If the cells are needed for further dissociation, then sorry. So the 525 cannot provide a further optimization or the further dissociation of your sample. So you have to simply prepare your cells going back into your BSCs. And then when the cells are optimal and ready for loading, then we will go on into the chip, loading into the chip. And then we have the isolation PCR hood for a sample-free, absolute clean environment for preparing master mix and preventing contamination. And we have a PCR machines and all the other equipments for the entire protocol. And lastly, we have the tape station system for sizing the library and also your cDNAs. So I would like to stop here. So there are some, I would like to show you in few slides for the sample submission form. So it is already available. So I will post it. So the link is here. So first of all, the user has to say that whether you are a SPS member or not, as the price are different. And you have to put on your personal particulars and also your sample nature, so which is an important field to fill up. So you need to declare that whether your sample is free from mycoplasma and also the MLV contamination. So we do not accept any cells or tissues being contaminated. So you have to talk to me before preparing the cells. So if you are working on mice, then you have to simply have to submit a report for a MHV negative report. Then we have to stay clear about this, that your mouse or your samples or your tissues are free from this one. And lastly, then other pathogens such as recombinant DNA and infections agents free. And also whether this is a genetically engineered and transformed virus. So all these things you need to declare. When you're through all this, then you need to declare your sample, your preparation, how exactly you're going to prepare your cells, because this is very important so that I can have a more idea, better idea to get known your samples, to prevent clotting of the chips and your cells, and to waste your valuable cells. And lastly, you have to declare and sign. And when you submit, then there will have a copy to me. Then I will send it to your PI for confirmation of your experiment. Okay, I think that's it for now. So I will take questions. Thank you.